Welcome to this talk on imaging workflows using Temporal to cure cancer and other useful things. It's going to be a bit like what we've done with Temporal rather than how to use Temporal. So I'm going to kind of just give you a tour de force and all the different things with which we've used Temporal. But first, you can't escape a micro CV. So I'm a senior manager of computational science. On the technical side, I use Java, Python, and TypeScript, and worked in something called computational fluid dynamics in the 90s. Worked in a web startup around finite element codes and computational fluid dynamics. And then worked in data acquisition for the UK's largest science project, Diamond Light Source, for almost 10 years. And then worked with some machine learning for an oil company called Halliburton. And then last 4 years, I've been working with genetic analysis at Jackson Labs. So that tells you I'm kind of a technical guy, kind of doing analysis, not necessarily as much on the software side as some people. I'm somewhere between a data scientist and a software engineer, I guess. Jackson Laboratory. This slide is entirely to prove that we have a nice logo, and I'll move on. So Jackson Laboratory is funded by mouse models, which means selling mice to researchers, and research grants. So people like the National Institute of Health would, give us a grant in order to do research, and philanthropy. So a lot of people will have been touched by the rare diseases, the cancer, and the different diseases which JAX helps to research, and they want to give something back. So there's this kind of philanthropic aspect to JAX as well. Our mission, put my glasses on to read it. Jackson Laboratory is a non-profit biomedical research institution discovering precise genomic solutions for disease and empowering global biomedical community in the shared quest to improve human health. And I wanted to read that out because that tells you everything you need to know about our approach to software and Temporal as well. Is that it's not like a commercial enterprise. So a lot of the time we'll be making research-grade software and then trying to get support the research community in what we do. So producing a scientific paper, producing reproducible software around that. So that's really important to understand. That's a very different mindset to what some people here might be used to. So and it can mean a smaller user group and maybe for each software maybe more bespoke. Our sites are, I'm based in Maine, and we have a large office in Connecticut, JAX Genomic Medicine as well. So we are a Cancer Institute designated center, and as well as rare genetic diseases, as well as looking into Alzheimer's and many different human diseases. The cancer part of our research is, a very important strand with more than 50 people employed. And another really kind of thing we're famous for is education. So there's some interns that come into the lab over summer, and I'm going to show one of their bits of work they've done, what somebody did for us this summer. That's a great opportunity for them and for us to share knowledge and to increase education. So that's kind of who we are, and that's our mission. So why do we analyze data? Why do we use Temporal? I'll get onto that, but why do we analyze data in general? Because it might be a research grant requirement. It might be required by a collaboration. We also have internal products and services which we want to support, and we also make new commercial products from time to time. And what kind of data is it? It's tabular genomic data, so columns of experimental results. And microscopy images we're going to talk about today. So those are, principally, we're going to talk about microscopy images. So those are produced by very high resolution instruments. So they have a kind of issue of scale, which I'll talk about. And Temporal helps with that. And the context of the analysis is that individual laboratories within JAX will have long-term research and postdoc members working on different projects, and we'll be supporting them. And then beyond that, they then have research-grade software, which is where we link that to scientific papers. We make the data fair, so findable, accessible, reusable, and interoperable, so that then is supporting the scientific papers. So there's the idea of reproducibility within research-grade software, and obviously we have a certain amount of commercial software as well. So history of durable pipelines. So I went to a great talk this morning from Diane, who, I don't know if you're in the audience, but she did a great talk on how they took their pipeline and then modified it and put it into Temporal. And we've obviously tried to do that too with previous products. We've used this list of technologies, if you're familiar with them, are not like replacements for Temporal, or even they are doing different things, but we've used them in that context. So Nextflow is this wonderful declarative workflow tool which allows you to specify inputs and outputs of all your scientific analysis, and then it will figure out what runs in parallel and what doesn't. So it's a bit kind of like the opposite to Temporal, where you pin everything down. Instead, you just declare roughly what you want and say to Nextflow, off you go. So that's one way that we've done kind of pipelines. Maybe not so much durable, that's why it's inverted commas. And then obviously we kind of rolled our own, built our own RabbitMQ, Redis, the key value store with events and things. And then in other organizations, I've also had the same problem. So using ActiveMQ and Kafka have also been another way of solving this problem. But actually, if we take a step back for that, and what are the features we want to be able to start and stop analyses. We want to be able to monitor progress, including failure states and cancel states. And we want, I think one of the key things with image analysis, as I'll get onto, is scale. So we want to be able to run things in parallel. We want to be able to tune memory. And we want to integrate with Kubernetes. We want to utilize the cloud for this analysis. It's not, the days of someone analyzing this on their laptop are gone, and even like a workstation now would have to have hundreds of gigabytes of memory just to process one image. So I'll get onto that, but instruments resolution have increased way beyond the desktop computing power, at least where we are today. And we need to be tolerant to resource downtime. This is a research organization, so databases might get updated or they might not be there. So Temporal has a wonderful durable nature, so that's an advantage for us. Multiple tech stack support, so none of our stuff is going to be on one tech stack. It's always going to be like Python stuff, there's going to be Java stuff, there's going to be, there's actually some Julia stuff in what I'm going to show today. So the science teams will produce that in whatever language, and then we have to figure out how to encapsulate that into an image processing workflow. And a strong community, it helps, and good adoption. Like I was very impressed by the kind of hockey stick curve that Temporal was showing off this morning in the keynote. So why did it get adopted so quickly? So what was it on day one when we used it that we thought, oh, we might like to try that? Obviously, there's the great tutorials. It's easy to get something running on that first day, and I'm going to show you that in the next slide. What we got going, and then what we turned into a product. The architecture is quite complete. So I spoke about those. Our kind of handmade versions of this kind of concept of durable workflows, and they've thought of many features that we didn't have, right? So that complete architecture is useful because eventually you're going to get onto that. So these are people who've solved this problem before multiple times and have then encapsulated it into a product which we could reuse. We like the fact that it was clearly easy to see how it scaled. It was a distributed system. You could see how these different Docker containers could be started and then orchestrated together. Configuring everything by code makes it easy on day one. You don't have to learn and go into some obscure YAML file or something. You just got it all right there. So that was just a really great experience of getting to run. And the other reason we adopted it so quickly is we didn't know the programming model was going to be so hard to learn at the start, right? So if anyone who's a bit more experienced with Temporal and getting onto the more advanced things, you don't see that necessarily when you first try it out on day one. So that's kind of like perhaps a negative thing, but also it's there for a reason. So our first worker was based around something called meta-analysis, which is for the mouse phenome database, which is a very long-running piece of software in the research community with reasonably large user group for our kind of domain. And it's deployed on Kubernetes. It's Java-based, and we wrapped that, and we're able to run that relatively easily and quickly. The runs last tens of minutes and produce millions of data points, which are then pushed back into this plot here that you can see, which is a Manhattan plot useful for researchers looking for signal in genetic data. So that's a kind of relatively simple use case. So we had this kind of initial prototyping, and then we solved our first product with it. So then we wanted to move on to a much bigger problem, which is image analysis. So we wanted to look at our image analysis in a more holistic way. So we had lots of different researchers making lots of different separate bits of code based around OpenCV or based around their own bit of desktop or maybe an ImageJ macro. Has anyone here heard of ImageJ? Hey, 2, 3. There's QPath, which is another one similar to ImageJ. People will have heard of PyTorch and things. But anyway, all these kind of stacks existed. And obviously that's replication, right? That's obviously, we're not replicating, that's duplication. We're duplicating many bits of effort into separate projects, and we want to bring those together into a single thing. And of course, the file size, as I mentioned, is an issue. You know, these huge files take a lot of memory and a lot of compute resources to analyze. So you know, that's another kind of string to this. So we knew because of that, we knew we wanted to leverage cloud. And obviously it's got the scale, it's got the orchestration with Kubernetes. And an S3 type system is going to make it cheap to have those instruments push their data into a cheap place for storage, and then we can analyze it later. And you know, some of these instruments can run pretty fast. In fact, it's a challenge just to save the data in the same rate that they can take the data. So it backs up onto SSDs and all sorts, but just to get that up and backed up and saved in the cloud is actually a computational challenge. But anyway, we're looking at analysis here. So the design, which we came up with for that, is basically to try and consolidate some of the front-end operation for image analysis in a nice Angular application, and you know, not try and reinvent the wheel, but do enough there that we don't need to keep reproducing lots of kind of front ends for different analyses. And then have a Spring Boot server talking to that and looking at the images, and then giving back images to the front end for the user to choose which analysis they want to work, and then encapsulating all of the image analysis into individual temporal workers of different types, which I'm going to go into and show you some pictures of what they do. So first of all, just a quick look at the user interface. It's Angular based. It's got you can view images and you can run image analysis jobs with it. It's got a scalable zoom, a bit like Google Maps or something like that. So it loads different resolutions from the underlying images because the images are so huge that as you zoom in, you want to go back and get the next lot of pixels and show that to the user. It's based around existing open source APIs. One of them is Plotly. There are other front-end JavaScript libraries that we put into that. And it basically connects to the server and has a relatively small user base. So large number of users is not an issue for our product. And it intentionally doesn't do everything. There's many image analysis front ends for selecting data and changing filters and all this, and everyone's got their own favorite one. So we're not trying to reproduce that. We're just trying not to repeat ourselves in all of these tools that we're deploying in Temporal by making a single web front end. And then the back end itself is doing that kind of Google Maps type ability where you use a library called bioformats. Has anyone heard of bioformats? So bioformats is like a library that allows you to read any microscope and other image formats. And there's a whole, as you can imagine, there's a whole load of vendors with all of their instruments. And bioformats is like a library that the open source community tries to keep up to date to read all of those file formats. And some of those file formats are like what's called pyramid files, where they'll have a very high resolution image and then a stack of lower resolution images going up, because it's so hard to interact with these data. The data are so large, they don't fit in memory. And the other great thing about it is it's got role-based access. So if you've used S3, you can put users onto different buckets, and then that gives us the ability for different research groups to have images in different buckets, and then we can just leverage that in the GUI by just showing the buckets as folders, and they can only see the images that are part of their group. So that gives us some kind of basic role-based access. And it's a microservice, and it's stateless and scales in Kubernetes. Okay, so on to the different temporal workers. As I said, they're a really heterogeneous kind of stack of things. We've got something called a slide cropper is based on Java and ImageJ, and then there's something called extending depth of field. I'm going to talk about these in more detail, based on C++ and Python. And you can go kind of down that stack, and there's always something a bit different Python versions, different libraries that the researchers have used. But of course, none of that matters, because we're going to encapsulate that into a Docker container with the correct libraries, and then we're going to put it as a temporal worker, and then we're going to talk to that to talk to those queues, using the server. So we don't mind what fruity stuff they used when they made the analysis. We just figure out how to build it. Now, one of them, the Julia one, was hard. Okay, making that Docker container was hard. And I know my colleague Nick, who did that work, was scratching his head for a while to figure out exactly how to build that into a kind of reusable thing. But I'll show you the results in a minute. Just as a kind of how the workers are working, they mostly follow a similar paradigm. So at the top there, you've got like a GUI that I said where we submit onto a server, and each worker has its own queue, but the queue, the worker has like the same state that it responds with, so like a standardized way. Okay, they all go queued, running, complete, or whatever. They go through the same state machine, and we use the temporal query to get the current state of the running worker and show that to the user, and we back that up. We actually back that up with our own database, so we have our own table of all the states of all the workers, and that's what the user sees. And we don't show them the temporal GUI. The temporal GUI is just for support engineers. It's a great thing, and we would like to show it to the users one day, but it's not quite aligned with the application that we've got. So, and in terms of an individual worker, it will basically follow something along the lines of downloading an individual workflow, having an activity to download one of these image files, and then some kind of analysis, probably some kind of parallel analysis involving multiple instances in Kubernetes, and then some kind of upload at the end. And obviously, you can straight away see that you're going to need some kind of shared disk for the way we've designed it, because that huge image is you're not going to want that to keep downloading that. So we have a shared disk between all the workers of a given type, and then they use the workflow ID from Temporal to store that image, and then if it's parallel, we'll all go back and hit that one image on shared disk, and that's just using features within Google, because we're using Google Cloud. So the first worker I wanted to show you is called Slide Cropper. I don't know if you can see the yellow boxes around each slice. Can you see the yellow boxes? Yeah, exactly. Put your glasses on. Me too. Well, what happens with this is that it runs that first step at a lower resolution. Remember I said that the image files are in like a pyramid, and it goes and grabs one of the lower resolution layers from that pyramid. And then it goes and finds, using a thresholding algorithm, it goes and finds all of the eyeball slices on that image, and then draws a box around them, and then produces those boxes as GeoJSON. So you have some kind of coordinate system. And then all it does for the parallel part of the workers is for all, I think there's 20 or 21 eyeball slices there. It'll run all of those in parallel at the full resolution and go and extract them and write them to their own separate images. So you've got from one huge slide image, you've got a bunch of individual tissues with which you can do further analysis. I'm going to show you some of the further analysis, but this is kind of the starting point. In terms of the code, this is a simplified view of the code, but you can see that find method then calls the find activity, and then it goes and finds all of those regions, which are using a thresholding algorithm to find all of those parts. And then it goes, for each of those, it's using an async, it's kicking off a temporal async function, so running all those in parallel. And then if the If the number of workers gets swamped by CPU or memory bound, then we rely on Kubernetes then to scale that out and carry on processing that parallel pipeline. And then you just wait for those promises on the Java side. So another worker is image alignment, and this is basically 3 images of the same tissue type. I think this is a slice of a kidney. I'm not actually sure. But basically want to overlay those and figure out a coordinate system which will allow them all to be rendered into one image so that researcher can compare that data in a likewise way. And you can see that one of them has been rotated. Well, it will actually rotate it back automatically and overlay it and give them a result that you see there. And that's written in OpenCV in Python. So obviously then we have to use the Python temporal clients. This is where having multiple languages is really helpful. And we're using the async stuff in Python there to just here we've got the download, and then we've got the run alignment, which is like a method taking 3 files, and it goes. It also takes a bunch of configuration parameters that the user has to set. So onwards and upwards. The next one is an extended depth of field worker. So what that does is if you imagine a microscope slide with a bunch of cells on, it's quite difficult to get them all in focus in one go. So what the microscopist has to do is scan down that 3D structure, and each one of the images that they take is going to only have a narrow band in focus. So can you see there that there's kind of a diagonal line of cells in focus? So what this tool is then doing is looking at that stack, processing them all, and then adding them up into one in-focus image. So that's kind of pretty cool, and it's pretty useful for researchers who just want to look at, actually kind of get a feel for their sample in a single image. It doesn't colorize it. It also works with color, so I actually have a color result and a gray. But if you put in a stack of images, you get a gray result. So another worker we use is using weakly supervised machine learning based on, to classify tissue age based on an ordinal classification model. And that's the one written in Julia that I mentioned, which is quite hard to make the Docker container, for which it was hard to make the Docker container. And what it does is it takes a bunch of known, it trains on a data set with a bunch of known ages of those cells, and then that's your ability to estimate the age of a given cell, because you've trained the deep learning on that. And then if in this case it would be a mouse who's been exposed to maybe cocaine or alcohol or high-fat, high-sugar diet, they're going to then get parts of their, this is a kidney slide, they're going to get parts of their kidney aged prematurely. So then you can run that same slice through the machine learning analysis and get like a heat map. So what you're seeing on the right-hand side is a heat map of all of the prematurely aged cells in that kidney sample. So that's what the red areas are. So then the researchers can use that to figure out where their experimental conditions have had an effect within the cells of that tissue. And that we think that's got a huge. We think machine learning in general, like everybody in the world at the moment, we think that it's got a great potential. So I'll show you a couple more examples with deep learning. So this is back to the eyeball. And you'll see in the retina that there's different layers of cells, and they all do different jobs. Don't ask me what they do. I'm not a biologist, but I do know that they exist and they do different jobs. And so what we want to do is extract and quantify those. So we want to use machine learning to do that, because that is very hard to write a general purpose algorithm to extract or to segment that image. And this one is written in Python. I don't know whether it uses TensorFlow or PyTorch. I think it's TensorFlow. And what you see here is the machine learning. Obviously, it's been trained on sections of eyeballs. And then this is some of the results. And you're seeing 3 masks for each of those 3 layers that has been generated by the machine learning on each of these 3 test images, which are actually quite different. And that mask can then be used in conjunction with the original image to pull out those layers of cells, and then those cells can be counted using an automated approach, or they can be quantified in other ways. But you have to first get the mask, which tells you which cells belong to which layer. So that's what Sonia was doing when she made this temporal worker. So another one is in general purpose object detection. I'm not sure if you can see, there's a tiny red rectangle down there in the bottom of the eyeball slice. I would ask you what it is, but you can see from the title, it's the optic nerve. So no prizes for that. But so the machine learning goes into that eyeball slice and finds the optic nerve for the researcher, and then is able to extract that. And, well, in fact, in this case, it's annotated it, and we've blown up the annotation. This, the guy who did this was one of our students, one of our interns over the summer. He's actually going to stay on until December. He had a lot of fun doing this. He sent me what he would call a low-res image, which I needed to use a special tool to download in order to put it in this slide. And then when I cut it out to put this, the second view, there was still way more resolution than I needed for the slide. So that it just gives you an idea of even the downsampled version of these high-res images is way bigger than you would need for something like a presentation. So that's it on the workers. I'll just wanted to talk a little bit about what we're doing to make this easier for research groups in future to make their own temporal workers. So we're using this tool called Cookie Cutter. Has anyone used Cookie Cutter? Yeah, one. So it basically like a templating tool. So it allows you to like predefine your temporal worker and then have somebody run a wizard, and then they end up with all the code, all of the YAML for deploying the worker, the Docker file and everything like that, because they don't know that, right? They only know the image. Well, they don't only know the image analysis, and there are lots of other things, but they mainly know the image analysis part. So we want to try and make it, as software engineers, we want to try and make it really easy for them to then plug that in. So we have this kind of cookie cutter design, and it hopefully avoids some of the temporal learning curve, because we just say put your analysis here, and we do the download bit and we do the upload bit, and they just do the kind of work on the files bit. So that's the plan there. I was thinking of, how are we doing for time? I was thinking of doing, okay, I'll move over this bit, but there's a couple of other workers outside of image analysis that I wanted to talk about for genetic analysis and machine learning there, but I'll skip over those. There's actually a diagram, slightly different architecture we've done for that. So future work in image analysis, we would like to do much more object detection. We've got more projects planned, and kind of almost make it into a production line, make lots of different object detection models working with different labs at Jackson Lab. And we'd like to use vision transformers. We haven't actually deployed those to a worker yet. We've got some prototype code. We'd like to do more in the open source area. If anyone's interested in actually seeing what we've done, in any context, even if you're outside of the kind of science world, we'd be very, very happy to open source a worker if we haven't got it open sourced already, and to figure out a way of showing off. People want to actually show how we've done this. I'm not sure it's very groundbreaking. I think you can probably look at the diagram and see roughly what we've done, but anyway, it's kind of interesting. And we would like to probably move to software as a service. If we had an internal deployment for the last 2 years, and we kind of had a more professional DevOps-y version in March that we're using, but then upgrading that and keeping it up to date and things is, it's got a cost attached to it, and we felt that cost is probably larger than the cost of the software as a service, but it's difficult to quantify all those costs, but it looks like a better option for us, I think. So in conclusion, the workflow pattern is not hype. We found it to be very durable. I think everyone's saying that here today. Easy adoption, good examples. The UI is great. We don't give it to our users, but it's great for diagnosing things. We've seen a lot of screenshots of it here today, people showing it off. And it's a very useful feature. On the perhaps not so positive side, the programming model learning curve is really steep, and you can make mistakes which have effects later on. They're not initially apparent. We certainly had that anyway. And doing your own deployment was pretty hard. So, I think on balance, we went down that route, and then we're probably going to stick to it because it's working and it delivers on the things it says. And like some of the keynotes today, where they were talking about trust, we definitely have that. We have the trust with the system, but that doesn't mean that there aren't some slightly hairy things there with the way you have to program it, and certainly some learning curve there. And thanks, basically, there's a whole load of people I'd thank, and a lot of the labs on the right have used this model, maybe only once or twice. So we've listed everyone who's been involved with it, but and a lot of the developers and scientists are listed there as well. So thank you very much. Do we have time for questions?
We don't, actually.
Yeah, any questions? Yeah, Diane.
So I imagine there's a lot of regulatory concerns here because you're dealing with a lot of medical device components. So how has that changed once you started moving, especially around deployments?
So the question is, there's a lot of regulation around this, and how have we dealt with it? Well, at the moment, we're mostly dealing with mouse data and then inferring that in human. So we haven't had to deal with the HIPAA part of it yet. So we don't have any, basically, is the short answer. Yeah.